Awesome. All right, so thanks, Bill, for introducing everything. Like he said, today I am going to present the work from my dissertation, which was titled The Test of the Effects of Geography, Gear Type, and Culture Techniques on Vibrio Risks Associated with Farm-Raised Oysters. So for today's seminar, I'm going to start with a short introduction to oyster aquaculture and resubmersion. Then I'm going to talk about the different factors that I tested and their effects on Vibrio recovery times. And then I'll wrap things up with a short conclusion for y'all. So to start, I wanted to introduce you to oyster aquaculture and seafood safety. Oyster aquaculture is a relatively new industry in Alabama. The first operation was established in 2012. Since then, the industry has continued to grow with a reported 22 farms operating as of the last report in 2018. Annually, Alabama farmers produce about 2 million oysters that are mainly consumed on the half-shell market. Farmers earn about 30 to 70 cents per oyster with an average price of 46 cents. There are 64 acres of water permitted for aquaculture with 37 acres currently in production. And farmers use a variety of growing methods, including off-bottom culture gear, single-set oysters, which we call oyster seed, that they can purchase from hatcheries, as well as routine handling practices that will produce a higher quality product. Now, off-bottom gear is used to suspend or float those oysters in the water column, as opposed to growing the oysters on the bottom like in a traditional wild reef setting. The two most common gear types are floating cage systems like the OysterGro system and the adjustable longline system. So the floating cage system floats at the surface of the water using two air-filled pontoons. The oysters are stocked into bags that will slide into that cage below the pontoons. The oysters can be held in the feeding position where they are submersed in the water. And then the cages can be flipped 180 degrees up into the flipped or desiccating position, allowing the oysters to be air dried. This is opposed to the adjustable longline system, which consists of a series of pilings and PVC poles, along which a horizontal longline is suspended. Oysters are stocked into baskets that are hung on the line, and this system allows for the oysters to be suspended at various steps in the water for feeding or raised in the air for desiccation. Both of these gear types are commonly used by farmers in the Gulf of Mexico. Because of the gear design, farmers can access their oysters at any time, which allows them to handle their oysters to improve their product quality. Two common routine handling practices are desiccation and tumbling. As I showed y'all, the gear is designed for desiccation, which involves air drying the oysters and the culture gear for up to 24 hours at a time. The oysters can survive that aerial exposure, but biofouling organisms like algae and barnacles can't. So by desiccating the gear and the oysters regularly, the farmer can keep them clean of biofouling. Additionally, farmers can tumble their oysters through a rotating mechanical grater. This grater is essentially a giant rock tumbler with various size holes. So when the oysters are sent through the grater, which washes the oysters and sorts them by size, that tumbling action will also chip off the fragile shell growth along the edge of the oysters, causing the oyster to grow with a deeper cup that is more desired on the half-shell market. So overall, off-bottom culture methods have several benefits over on-bottom culture methods. By raising those oysters into the water column, the farmer provides greater protection from predators and reduces mortality rates. Additionally, the oysters have greater access to food in the water column, which can increase their growth rates. And finally, the culture methods allow the farmer to be able to access their oysters at any time, allowing for those routine handling practices that will keep their gear clean and also produce that perfect, clean, deep cup oyster for the half-shell market. Now, oysters are commonly consumed raw, which presents a public health risk from the bacteria naturally found within oysters. So oysters are filter feeders, and they will naturally concentrate the Vibrio bacteria that are found in waters where oysters are grown. Unlike other foodborne pathogens that you think of, like Salmonella or E. coli, Vibrio bacteria are naturally found in the environment, and so you can't see them as a source of contamination within oysters. As a result, oysters are regulated to ensure their safety as a food product, and there are 2 species of Vibrio that are considered a public health risk when oysters are consumed raw. The first species is Vibrio vulnificus, or VV. VV will infect those who are immunocompromised, resulting in gastroenteritis and primary septicemia. VV is an important foodborne pathogen because it accounts for 95% of all seafood-related deaths. The second species is Vibrio parahemolyticus, or VP. VP can infect anyone and has been known to cause widespread outbreaks in the past. VP infections result in gastroenteritis and septicemia. VP is also an important foodborne pathogen because it is the leading cause of seafood-related infection. Now, Vibrio risks are important to consider during the oyster farming process. When a farmer removes those oysters from the water for routine handling, there are Vibrio risks that can be associated with those activities. Since Vibrio bacteria are naturally found in waters where oysters are grown, there is what we would consider the ambient or normal levels of Vibrio within the oyster. So in this graph here on the right, you see the Vibrio levels on the vertical axis and time on the horizontal axis. And that dotted line represents those ambient levels within the oysters. So before handling, the oysters are in the water at the farm site and they're actively filter feeding. So the Vibrio levels would be at the ambient or normal levels within oysters. Then the farmer takes those oysters out of the water for handling. And during that handling period, which is typically up to 24 hours, they are no longer filter feeding. The oysters can also be exposed to higher temperatures outside of the water, which creates the perfect little incubator for Vibrio bacteria to grow. After the handling period, oysters can have significantly higher Vibrio levels than before the handling period. So after the farmer is done handling the oysters, they are put back into the water at the farm, or what we call resubmersed, and the filter feeding can resume. During what we call a resubmersion period, the oysters will purge the higher levels of Vibrio bacteria. And after an appropriate resubmersion period, the elevated Vibrio levels have recovered back to the ambient levels found in oysters that were never taken out of the water. From there, we considered the elevated risk imposed by those handling practices to be mitigated, and the oysters can be harvested within an appropriate time to temperature window and consumed on the raw market. So in 2013, shortly after oyster farming started in Alabama, the Alabama Department of Public Health required guidance on the resubmersion of cultured oysters. At the time, the best available data was from Alaska, and a 30-day resubmersion requirement was put into place. So in 2015, the Interstate Shellfish Sanitation Conference funded a study to further investigate resubmersion in Alabama. And this study looked at the Vibrio recovery times for oysters that were desiccated and then resubmersed in long-line baskets underneath a pier, which was built to mimic the conditions on an oyster farm. This study found that 14 days of resubmersion was sufficient for those Vibrio levels to recover back to ambient levels, and the requirement was reduced from 30 days to 14 days. However, the industry was still concerned about biofouling organisms reinfesting the oysters within 14 days, as well as the potential for rainfall closures that could prevent harvest while they are waiting out that resubmersion period. So a follow-up study was conducted by Stephanie Grodeska. She was a former graduate student under Bill Walton at the Auburn Shellfish Lab, and she used our research farm site in Portersville Bay, Alabama. She looked at the Vibrio recovery times in desiccated oysters and found that 7 days of resubmersion was sufficient. So using this data, the Public Health Department further reduced that resubmersion requirement from 14 days to 7 days, but only for those farmers that were using desiccation in the adjustable longline system. So currently, the resubmersion requirement in Alabama is 7 or 14 days, depending on the gear type and the handling type that the farmer is using. But between those two resubmersion studies, scientists and public health agencies have questioned whether there are other factors that should be considered when determining resubmersion requirements. So that partial reduction revealed several knowledge gaps surrounding resubmersion. There are 4 factors that have yet to be tested for any potential effects on resubmersion. There's the handling type that's used by the farmer, the culture gear type being used, the time of year that the handling and resubmersion is occurring, and the geographic location of the farm. So the overall goal of my research was to investigate these factors and provide data for the regulatory authorities to make more informed requirements. So we performed a series of 3 resubmersion studies at 3 farm sites in Portersville Bay, Alabama, Grand Bay, Alabama, and Cedar Island, North Carolina. For today's seminar, I'm going to show you the data by factor. So this table kind of gives you an overview of the factors that were tested during each of the 3 studies. Portersville Bay was the very first study that we ever did, and that kind of dived into the effects of different handling types. But while we were doing this study, it prompted the other 3 factors that we wanted to look at, which is why those factors were later tested in Grand Bay and Cedar Island. So first, let's talk about the effects of handling type on Vibrio recovery times. So all of the previous research has only looked at the recovery times after oysters were desiccated, leading to that partial reduction in the requirement for farmers. However, farmers use other forms of handling as well, leading to a need to investigate those handling types. So tumbling is another common handling type. Compared to desiccation, it is a rougher form of handling, as the oysters are tumbled through a giant rotating metal tube. So that tumbling has the potential to create additional stress on the oysters, which could delay the resumption of filter feeding after the oysters are resubmersed. So if this is true, then tumbled oysters could possibly need a longer resubmersion period than desiccated oysters. Additionally, the idea of using an overnight refrigeration period instead of an overnight desiccation period was proposed. Traditionally, when oysters are desiccated, they are exposed to higher air temperatures that allow those Vibrio bacteria to rapidly grow. Instead of air drying the oysters outside, they could be air dried in refrigeration, which is known to prevent Vibrio bacteria from multiplying within the oyster. There have been some previous studies in New Jersey and Alabama that took oysters that had been refrigerated and resubmersed them in the water. And they found that the Vibrio levels actually increased immediately after resubmersion before recovering back to those ambient levels. So while these studies were not performed specifically with the intent of examining the effect of handling practices in an aquaculture setting, they did provide some preliminary findings that we wanted to further investigate. So the effects of handling types were examined at all 3 of our study sites. We had 2 research farm sites in Alabama and a commercial farm site in Cedar Island, North Carolina. During these studies, we tested up to 4 different handling treatments with the submersed control. The submersed control oysters remained in the water during each trial, and that allowed us to measure the ambient levels of Vibrio within the oysters. The oysters in the 4 handling treatments were removed from the water, and half of the oysters were tumbled or sent through a mechanical grater once. The other half of the oysters were not tumbled, and they just remained in their bags. From there, we took half of the tumbled and half of the not tumbled oysters and placed them into a walk-in cooler for an overnight refrigeration period, while the other half of the oysters were not refrigerated. Instead, these oysters remained outside for desiccation. So this design yielded 4 different handling treatments. We had tumbled and desiccated, tumbled and refrigerated, refrigerated only, and desiccated only. So the full set of treatments was tested in both Portersville Bay and Cedar Island, but in the Grand Bay study, the only treatments tested were the tumbled and refrigerated and desiccated only treatments. So each resubmersion trial was 2 weeks long, and it consisted of a series of sampling days. That way we could capture those changes in Vibrio levels over time. So before we did anything to the oysters, we collected pre-treatment samples. Then we had the treatment period where we pulled the oysters out of the water and applied those treatments. After those treatments were applied, but before resubmersion, we collected our post-treatment samples, and then we resubmersed the oysters in the water. Then we had a 14-day resubmersion period, and we collected samples at various time points during those 14 days. That way we could measure the Vibrio levels to determine recovery. So back at the lab, each sample was tested for 4 different Vibrio targets. We had Vibrio vulnificus, or VV, total VP, as well as the pathogenic VP strains, which is both TDH positive and TRH positive. The Vibrio bacteria in each sample were enriched overnight using the 3-tube most probable number analysis. Each sample consisted of 12 oysters, which I aseptically shucked into a sterile blender and then blended it to create what we call an oyster homogenate, or an oyster smoothie, whichever you prefer. From there, the oyster homogenate was serially diluted in phosphate buffered saline, and the dilutions were used to inoculate a series of tubes containing a growth media called alkaline peptone water. After an overnight incubation, we took growth from each of the tubes that were turbid, meaning that bacteria grew, and boiled them to extract the bacterial DNA. Then we identified the presence of each Vibrio target and confirmed it using real-time PCR analyses. And the number of tubes that were positive for each Vibrio target were used to calculate the most probable number of bacteria per gram of oyster meat using the table you see below. Prior to analysis, all of the Vibrio data was log transformed for normalization. Then the data were analyzed using mixed effects models in R with the fixed effects of treatment, time, and the interaction between those two factors. And I also included a random effective trial to account for any environmental variation experienced among the trials. So this first set of graphs shows you the Vibrio levels in each of the treatments before and after the handling occurred, but prior to resubmersion in Portersville Bay. So you see the 4 different Vibrio types, VV, VP, TDH, and TRH. On the horizontal axis is each of the treatment types, and the vertical axis shows the Vibrio levels in log NPN per gram. So first, you can see that the refrigeration treatment was effective at preventing the Vibrio from growing in all 4 Vibrio types. We did not see significant increases in Vibrio levels within those oysters, while the non-refrigerated or desiccated oysters had significant increases in all 4 Vibrio types, in some cases by up to 1 to 2 log NPN per gram. So this next series of graphs will show you the Vibrio levels after resubmersion. You have VV on the left here and VP on the right. The horizontal axis now shows the days since resubmersion, and the treatment bars are grouped by day. So after 1 day of resubmersion, we saw a spike in the Vibrio levels in the refrigerated oysters, which is similar to what had been previously described. And we saw that all of our treated oysters had higher Vibrio levels than the control level, or the control oysters. After 2 to 4 days of resubmersion, we start to see the VV and total VP levels begin to recover back to ambient levels. And once recovered, they remained similar to or lower than the ambient levels in the control oysters. This next set of graphs shows the resubmersion data for the pathogenic VP strains, TDH and TRH. You can see that it took 7 days of resubmersion for those pathogenic VP levels in the treatments to recover back to the ambient levels. So this table summarizes the Vibrio recovery times from Portersville Bay based on the Vibrio type and the treatment type. The Vibrio recovery times ranged from 2 to 7 days, and the different Vibrio types recovered at different rates, with VV having the fastest recovery time, and pathogenic VP had the slowest recovery times. Regardless, the Vibrio levels in all of the treated oysters recovered after 7 days of resubmersion, similar to what was found previously for desiccated oysters. In this study, there seemed to be no effect of tumbling and refrigeration on the overall Vibrio recovery time. Now, when we look at the effect of handling type in Grand Bay, we saw a slightly different result. Just to remind you, in Grand Bay, we only used the tumbled and refrigerated and desiccated treatments, but we used both aquaculture gear types. And in this case, the tumbled and refrigerated oysters required 14 days or more of resubmersion for those Vibrio levels to recover, while the desiccated oysters only had a 7-day recovery time. So we still saw the same variation in recovery times among the Vibrio types, with VV recovering the fastest, and in this case, the total VP levels required the longest recovery time. Moving on to the results from Cedar Island, North Carolina. We saw similar results here as we did in Grand Bay. Just to remind you, this study tested all 4 handling treatments like the Portersville Bay study did. And what's interesting to note here is that the tumbled and refrigerated oysters required 14 days, while all of the other treatments only required 7 days of resubmersion for those Vibrio levels to recover. So in conclusion, The effects of tumbling were both site-specific and dependent on if it was combined with an additional refrigeration step. When combined with refrigeration, the tumbled and refrigerated oysters required 14 days or more of resubmersion, while when that tumbling was combined with desiccation, it only required 7 days of resubmersion. The refrigeration treatment was able to prevent Vibrio from growing during handling, but after resubmersion, those Vibrio levels actually increased before they began decreasing and eventually recovered back to ambient levels. Overall, refrigeration did not reduce the Vibrio recovery time after resubmersion. So these studies showed that Vibrio recovered after 7 to 14 days in all handling treatments, with some of the Vibrio types recovering faster than others. The tumbled and refrigerated oysters consistently required 14 days or more of resubmersion at 2 out of the 3 study sites, so it would not be recommended as a common industry practice. So now we're going to move on and discuss the effects of gear type on recovery times. So while a previous study has shown that the ambient Vibrio levels are similar between oysters in the adjustable longline system and OysterGro gear types, resubmersion research is strictly limited to the adjustable longline system. So in the absence of data for the floating OysterGro cage system, the resubmersion requirement was not reduced for those farmers, as the potential effects of gear type is still unknown. So when using the adjustable longline system, the oysters can be hung at any depth in the water column. And as a result, these oysters may be subjected to less wave action that would interrupt their filter feeding, allowing them to purge elevated Vibrio levels more effectively than oysters held in other gear types. This is in contrast to oysters in the floating cage system, which are held at a fixed depth right below the surface of the water. Since they are free floating, they can be subjected to longer periods of wave action, which has been shown to reduce the filtration rate of oysters. In this case, the wave action could affect how effectively the oyster can purge elevated Vibrio levels and may result in a longer recovery time. So this study was performed in Grand Bay, Alabama, using both gear types. We performed trials in 2 months in May and July of 2018 and 2019. And again, we only looked at 2 handling types, the tumbled and refrigerated and desiccated treatments. Similar to before, the same sampling scheme is used, but in this case, we used reduced sampling days during the resubmersion period. So for the sake of time, I'm not going to show you the pre and post treatment data from the study, but in both May and July, we saw the same pattern as before, where the tumbled and refrigerated oysters did not have that spike in Vibrio levels, while the desiccated oysters did have that spike. So this set of graphs shows the Vibrio levels after resubmersion in May during the Grand Bay gear comparison. The graphs are set up similarly as before, but in this case, the first 3 bars are grouped for the adjustable longline system oysters, and the second set of 3 bars represent the OysterGro oysters. So first, I just wanted to note that the ambient Vibrio levels in the control oysters that never left the water were similar between the 2 gear types. After the oysters were resubmersed in May, the recovery times for each Vibrio type ranged from 7 to 14 days. The Vibrio levels in all treated oysters recovered in 14 days, except for the adjustable longline system tumbled and refrigerated oysters, which recovered after 7 days. The next set of graphs shows the Vibrio levels in oysters after resubmersion in July. And you can see in July, the recovery times actually ranged from 3 to 14 days. So some of the Vibrio types recovered a little faster in July. All of the Vibrio levels recovered in all of the treated oysters with one exception. The total VP levels in the ALS tumbled and refrigerated oysters were still significantly higher on day 14, so the levels did not recover during the sampling period. So this table summarizes the recovery times by Vibrio type for each of the gear and treatment combinations. And when oysters from each gear type were desiccated, the overall recovery times were similar in both May, which was 14 days, and July, which was 7 days. When we get to the tumbled and refrigerated treatment between the gear types, it got a little more convoluted. In May, the OysterGro oysters required a longer recovery time, while this trend was opposite in July, where the adjustable longline system oysters required a longer recovery time. So this study found no apparent effect of gear type, as the recovery times were similar for both gears. In fact, the recovery time varied depending on the handling type instead, with those tumbled and refrigerated oysters requiring longer recovery times than the desiccated oysters. I just wanted to note, though, that this study had lower replication compared to other resubmersion studies. So I'm only discussing trends here, and they should definitely be further investigated. So next, let's talk about the effects of time of year. So all of the previous resubmersion research was performed during summer months between July and September. These studies were performed during this time because it is the peak of the high-risk season for Vibrio infections. But that high-risk period actually starts in May, and infections can still occur early in the season. And there are some differences in the Vibrio levels between early May and that July through September period that could have effects on the recovery of elevated Vibrio levels. So based off of the literature, we know that there are higher pathogenic VP levels in oysters in early May. Combine that with the fact that previous resubmersion studies have found that those pathogenic VP strains require longer recovery times than for total VP, indicating that the recovery times in early May could be longer for those Vibrio types. Additionally, early May is characterized by cooler water temperatures, which correlates to lower filtration rates in oysters, meaning the oysters might be less effective at purging the elevated levels of Vibrio after handling. This is in contrast to July through September, when there are higher VV and total VP levels in oysters. But we do know that those recovery times for VV and total VP are faster than they are for the pathogenic VP. So this time period also has higher water temperatures, which can result in higher filtration rates in the oysters and could make those oysters more effective at purging the Vibrio bacteria. So based on all of this literature, it is possible that the Vibrio recovery times may actually be longer in May than during the peak of summer. So time of year was examined at our study in Grand Bay, Alabama. Just to remind you, we use those two gear types for those two handling types, and we performed those trials in early May and in July. Again, the experimental setup is the same as before, but I put it here briefly to remind you of it. And first, I wanted to show you the environmental data from this study. You can see that our water and air temperatures were about 6 degrees cooler in May than they were in July, which is what we were aiming for. So I showed you the graphs from this study during the last section. So for the sake of time and to discuss the effects that we saw from time of year, I'm only going to show you the summary table of recovery times. So when considering each gear and treatment combination, you can see that the recovery times for desiccated oysters were longer in May than they were in July. The same pattern was not seen for the tumbled and refrigerated oysters, where the recovery times depended more on the gear type used in combination with that treatment. So in May, the OysterGro oysters required a longer recovery time, and the opposite was true in July, where the adjustable longline system oysters required a longer recovery time. So in conclusion, the recovery times for the tumbled and refrigerated oysters varied between May and July and sometimes required more than 14 days. So we didn't really see a clear effect of time of year with this treatment. Because of the fact that the adjustable longline system oysters did not recover when subjected to this treatment, we would not recommend this as an industry practice. When we look at desiccation, we did see an effective time of year. So the recovery times for desiccated oysters were longer in May than they were in July, potentially because of all of the reasons I stated before. However, this study, again, only had limited replication, so the trends can only be discussed here. And finally, let's talk about the effects of geography on Vibrio recovery times. So several resubmersion studies have been performed in Alabama waters as well as in New Jersey and Washington state. These studies have determined Vibrio recovery times after a variety of common industry practices, which are unique to each of their geographical locations, some of them using both wild and farmed oysters. So in Alabama, the studies that I described to you in Portersville Bay and Grand Bay, Alabama, these were done with oysters that were always subtidal, meaning they are always submerged unless somebody goes in and pulls them out of the water. These oysters were subjected to different gear and handling types, and the recovery times were 7 to 14 days, depending on the handling type used. In New Jersey, the oysters are intertidal, meaning that they are exposed to the air as the tide goes out and then resubmerged when the tide comes in. So those oysters were removed from the water and packed into harvest containers and then refrigerated overnight. After they were resubmerged in the water, it was found that the Vibrio levels recovered after 2 days. In Washington state, both cultured and wild oysters are intertidal and they are desiccated during each low tide. These studies found that Vibrio levels recovered after 1 tidal cycle in the water. Now, all of these studies tested different gear types, culture practices, harvesting techniques, all based on the common industry practices within each region. But as more and more of this resubmersion research is done, the industry has asked, how many resubmersion studies do we need to do before we can come up with an answer? So these studies are expensive and time consuming, and a lot of people have asked, can we apply the results from one geographical location to another geographical location? So to kind of test this, we wanted to know if there would be a geographical effect in oysters from different locations that were treated similarly. For example, Alabama and North Carolina farmers both use a similar gear type, the OysterGro gear system, as well as similar handling types of desiccation and tumbling. The oysters are also subtidal and raised out of the water manually for desiccation. However, there are differences between the states in terms of Vibrio ecology and environmental conditions, which could affect oyster behavior after resubmersion and the changes in Vibrio levels during handling and after resubmersion. So these factors could prevent applying the findings from a study performed in one water body to aquaculture operations in other water bodies. So to look at geographical effects, we performed 2 studies simultaneously. The first study site was located at a commercial farm site in Cedar Island, North Carolina. 4 resubmersion trials were performed in 2018 and 2019 using that OysterGro gear system and the 5 treatments that I have previously described. But I just wanted to point out that this study was designed to use common factors and treatments as the Grand Bay, Alabama study. So both studies used a similar gear type and similar treatments, and they were performed during the summer, allowing for us to compare the recovery times between North Carolina and Alabama. So the North Carolina study had a similar sampling scheme as previously described, but I just wanted to highlight that the North Carolina study shared common sampling days with the Grand Bay study. So to show you the results from this, this table will summarize the recovery times found in North Carolina. I wanted to point out that the Vibrio vulnificus, the VV recovery times were indeed only 1 day. The desiccated oysters did have a significant spike in VV levels after handling, but those elevated levels recovered after 1 day of resubmersion. This is in contrast to the refrigerated oysters that did not have a spike in VV levels after handling, and they did not see a significant spike after resubmersion. Therefore, the recovery times for VV were the fastest out of all of the Vibrio types. Meanwhile, the total VP levels had the longest recovery times in this case. And overall, the Vibrio levels in all of the treated oysters recovered after 7 days of resubmersion, except for the tumbled and refrigerated oysters, which required 14 days. When we compare the Vibrio recovery times between Grand Bay, Alabama and Cedar Island, North Carolina, we see similar trends. The tumbled and refrigerated oysters in both states required 14 days of resubmersion for those Vibrio levels to recover, while the desiccated oysters only required 7 days. The VV and pathogenic VP strains recovered faster in Cedar Island than they did in Grand Bay. And in both areas, the total VP strain took the longest to recover. Regardless, the overall recovery times were the same for the treatments in each area. So in conclusion, the Vibrio levels within the oysters experienced similar changes during handling and after resubmersion in both locations. We saw that VV recovered the fastest in both locations, and VP recovered the slowest, and the recovery times were similar for the treatments that were tested. When we expand the geographic scope and compare these findings from these locations to the findings in Portersville Bay, Alabama, we see that the desiccated oysters had similar recovery times of 7 days across all sites. But those tumbled and refrigerated oysters were a different story. The recovery times ranged from 7 days in Portersville Bay to 14 days in Grand Bay and Cedar Island. So there was some conflicting results there, as well as the limited geographical scope of these studies. And this indicates that there could still be the potential for temporal and geographic variation in resubmersion times, especially between growing areas and waters of the same state. So now just to wrap things up with a quick summary for the industry. We know that routine handling practices expose oysters to conditions that are conducive for Vibrio to grow within the oysters, and this can lead to an increased public health risk. But we know through previous studies and the research I presented today that resubmersion is an effective mitigation strategy for farmers. We found that several factors can affect Vibrio recovery times. Tumbling and refrigerating oysters produce the most variation in and the longest Vibrio recovery times. Adding that refrigeration step while the oysters are removed for handling did not reduce the overall recovery time and actually produced the longest recovery times when combined with tumbling. Therefore, we would not recommend this as a common industry practice. Overall, the recovery times for oysters ranged from 7 to 14 days, depending on the handling type, gear type, time of year, and location. Oysters that were desiccated or tumbled and desiccated required 7 days of resubmersion, while those that were tumbled and refrigerated required 14 days. And finally, due to sampling and time constraints, the studies that tested the effects of gear, time of year, and geography had limited replication. So future studies are needed to confirm the trends that I observed. Regardless, the work I presented here today has shown some interesting trends, and the data is available to the appropriate agencies. That way they can use it to inform existing regulations. And with that, I just kind of wanted to wrap this up to kind of give you an idea of what I've done in the past 4.5 years. I performed 13 resubmersion trials, and over those 13 trials, I processed a little over 1,200 samples, which means that I shucked over 18,000 oysters. And thanks to Jessica Nash, if you were watching, inoculated over 25,000 tubes. As Bill said, I was able to secure over $500,000 in grant funding to do this work. And that also funded me to travel to different conferences. So I gave 13 oral and poster presentations over 11 conferences in 10 states. I have 2 publications that just recently came out, with another 2 in the works. And I think my funniest achievement is I do have 1 tape ball inspired by the tape ball from the lab that I worked in undergrad. And that tape ball is almost 5 pounds. And to wrap things up, I couldn't have done all of this stuff without a lot of people involved in my life. So I just wanted to give a big special thank you to my co-advisors, Bill and Jess, as well as my committee members, Todd and Jim. All of the people at the FDA lab, thank you, whether you helped me shuck an oyster or gave me a snack or gave me some words of encouragement along the way. I also wanted to thank the Auburn Shellfish Lab staff and students for helping me with all of the field work. especially Glenn for taking me out in some of the worst weather you could ever imagine. I wanted to thank all of my funding sources that made this possible. And last but not least, all of the people in North Carolina. I know some of you are watching today. So Jay Styron of Carolina Mariculture let me use his farm, as well as the Noble Lab at the UNC Institute of Marine Sciences. They opened their doors and let me invade with my 47 boxes every summer. So thank you to everybody. And with that, I can take any questions.
Great. Thank you, Vicki. And while people can, there's a couple ways to do questions. We could have people just unmute themselves and ask, or I can read them out of the chat for Vicki as we go. I just also want to remind people, anybody that at Auburn's School of Fisheries, the announcement for this seminar had a Qualtrics link to provide some feedback to the department. So please take a minute and fill that out. So does anybody have any questions they'd like to ask Vicki? Or comments?
Hi, Vicki. Can you hear me?
I sure can, Carolyn.
Hey. Hi. I had a quick question about the resubmersion. Is it necessary if you do the refrigerated treatment? Because the refrigerated treatment seemed to keep the Vibrio levels practically at ambient level, and it wasn't until they were resubmerged that they spiked. So is there some other benefit to the resubmersion that I'm not thinking about?
So we were looking at pre-harvest techniques. Obviously, if a farmer is harvesting their oysters, they would put them into refrigeration right away. These may be oysters that are not quite ready to go to market. They're still a little too small for that. And so we were seeing if refrigeration could help prevent significant increases, as well as if those oysters are ready to go to market, if you refrigerate them and then put them back into the water, could it help speed up that process of getting them to market? So could you have a 2-day resubmersion period instead of a 14-day resubmersion period? In this case, it didn't reduce the recovery time at all. So that's why we don't recommend it as a practice.
But if you're at the harvest point, is it recommended? I don't know anything about oysters harvesting, but is it typical that you refrigerate immediately upon harvest?
Oh yeah. Oh yeah. There's what we call time to temperature windows. So depending on the time of year, it can be as little as 1 hour to get them to refrigeration. Sometimes in the winter, I think that's more. I think you have like 12 hours, but don't quote me on that. But yeah, definitely whenever they are harvesting, they try to get them to refrigeration as fast as possible.
Okay, thank you so much.
You're welcome.
Thank you, Carolyn. Any other questions? I know committee members may have questions, but I think we'll save those for after.
I see Todd nodding. I'm nervous.
All right, I'll give a couple more minutes. I know sometimes it's harder for people to.
There's no questions. I got a comment that Victoria now has the record for Zoom presentation attendance at 51. So congratulations on that, Victoria. That's quite a lot of people.
Well, thank you.
Yeah, so great work.
And only summer related. So, all right, so Brian, there are two questions for you, Vicki. So Brian Jones has asked, how problematic were tropical storms with the research?
Ooh, I do remember Tropical Storm Cindy. I think that was my first summer here. Glenn and I were watching the radar, and we go out in between bands of rain. Totally safe, 100% safe, but luckily I was able to skate around most of the storms. My research was done this summer, so that was good.
And then PJ has a very practical question. If you've shucked, I think you actually had 18,000 oysters shucked. So what is your recommended shucking knife?
You can use a screwdriver. I have done that before. I prefer a 3-inch Dexter. That's my favorite.
And the carpal tunnel syndrome.
That's right, yeah. I didn't put that on there, but I did get one new wrist out of the deal.
Okay. All right. Oh, and then Adrian has asked, have you thought about how you'd like to continue your research in Texas? Like, would you like to continue this or any potential next projects that you have in mind?
I know that the people in Texas already want me to do a resubmersion study to see what they need there. I think it would be interesting to see if there's any other things that they could do along the way to help make oysters safer. Definitely something that's an interest of mine is developing a more rapid assay. A lot of the assays that we use in the lab, they take a while. They're good methods, but they do take a while to get results. So if there's something that we could do in the laboratory to speed up that process and make it more accessible to farmers, that would be awesome.
All right. All right. I think with that, unless anybody has any last minute pressing questions, we're going to let you off the hook, Vicki. Thank you, everyone, for attending. Appreciate your attention. Please, again, if you're with Auburn, please do fill out that Qualtrics survey. We do need that information. Thank you. And Vicki will reconvene with your committee in a bit.
Sounds good. Thank you,
Thank you, everyone.
everyone.